Now we are going to discuss these pyrenoids. Pyrenoids are very special component because they contain an enzyme that is starch synthetase. From the name itself, it is going to be very clear to you that this enzyme is going to help in the synthesis of starch. This is going to help in what? In the synthesis of starch. This enzyme starch synthesis is going to help in the synthesis of starch. So this is very special component which is present in, see, this is an image of Chlamydomonas. Chlamydomonas has only one chloroplast, that is, you can see this cup shaped chloroplast, okay, and this chloroplast is containing this very special component that is known as pyrenoid. Towards this pyrenoid, you can see these are some starch granules. These starch granules accumulate towards this pyrenoid structure, and this pyrenoid contains an enzyme that is starch synthetase, which is going to help in the synthesis of starch. So this pyrenoid occurs in only few species, okay. Then we move on to the next, that is the stigma. Now the stigma is also known as an eye spot. Stigma is also known as what? It is also known as an eye spot. Now this eye spot is actually photosensitive. It is photosensitive. Since this eye spot is photosensitive, this is going to have some photoreceptor pigments. Normally what happens in a cell, the plasma lemma and the chloroplast envelope contains these photoreceptor pigments. As you have seen that this is Chlamydomonas cup shaped chloroplast, and here is one eye spot that is contained within this chloroplast. And when you look at this structure of this eye spot, what is going to happen? You can see these are the photoreceptor pigments present over here. These photoreceptor pigments are what? They are photosensitive. So they are going to help to actually receive light, okay. They are going to help to receive light because they are photosensitive pigments. So this stigma and eye spot actually occurs in case of only certain, you can say, motile stages of algae. Motile stages, for example, I have given example of Chlamydomonas. This eye spot occurs in case of Chlamydomonas, okay. Now till here I guess this envelope, matrix or stroma, thylakoids, pyrenoid, stigma, it is very clear to you, and this ultrastructure of chloroplast is very clear in your minds, okay. Next I am going to just give an overview of this structure, how you can draw it in your examination. So let us start with discussion of this overall structure we have discussed so far. We have talked about the envelope, right, which is made up of two membranes, that is the outer membrane and the inner membrane. See, the outer membrane and the inner membrane, and between this there is space, that is fluid filled space, which we also call as periplastidial space, intermembrane space, right, and chloroplastidial space also. Then we have a circular DNA. Generally it is circular, but it can be linear also in some cases. Then this is one thylakoid, okay, this is one thylakoid, and when this thylakoid are arranged in this shape, this discoid thylakoid is known as what? It is known as a granum, and this granum are linked together, this is by lamella over here, alright. And we have also in the matrix or the stroma, this stroma or the matrix contains lipid globules, this starch grains and ribosomes also. So this is an overall, you know, diagram. How you are going to draw this diagram in your examination? Remember, just draw the two membranes, because since it is a chloroplast and it is a form of plastid, it is going to contain what? It is going to contain two membranes. I am just drawing this diagram just to tell you how you are going to draw it in case of your examination.